class today, the Wednesday of 20th. Now, allow me to share my screen. So maybe before I share my screen, I would like to note that in our last or previous class, we were revising something about growth. And I do believe for the most content areas we covered. So in our class today, we will be looking at reproduction and not growth anymore, because most of the content areas we did cover in our previous class, most of the most examinable areas. I would also want to inform you that after this class today, you will get the materials that we used in our previous class, and of course in this class today. So feel most welcome, and karibuni sana. So I hope you can see me share my screen. So our questions today will be entirely on the topic reproduction. So we begin with the first question. What is reproduction? So I'd like to see responses for that. What is reproduction? Any hands up? What is reproduction? I can see Leon Lewis. I can see others. What is reproduction? No hands are up. Are you people here with us? Are you, or you're still sleeping? This paper is a procedural paper tackling reproduction in various places. So I can see Evans. Evans, you can unmute and speak.
It is the process by which mature living organisms give rise to young organisms. Sorry, I'm not sure.
Okay, that is a good trial. Now you can see as simple as just definition can be a total mess to us. We need to know how to define these terms. And a good biology paper cannot miss such definitions. That was good, Evans. That was a very good trial. So I would ask someone else to answer that. Yes, Michael, you can unmute and speak.
Reproduction is the process by which living organisms give rise to new individuals of their own kind.
That is very good. That is how we define reproduction. So it is that process whereby living organisms will give rise to young one of their same kind. Let us please note that for all the 8 characteristics of living things, they are processes. None of them occur within a twinkle of an eye. And as such, it's good that when we define these characteristics of living organisms, we start with the word process. So it is a process whereby living organisms give rise to young one of the same kind. And most often, we tend to define reproduction as species. And those terminologies are very, very different. And I would say for species, we define them as the smallest unit of classification whose members can freely or naturally interbreed to produce a fertile or a viable offspring. For reproduction, it does not need to be viable. It just must be of the same kind. So please let us note that specific difference. So give 2 reasons why reproduction is important. Why would reproduction be important to living organisms as a characteristic. Yes, I can see 26917. You can unmute and speak.
It leads to quality improvement.
That is one. It leads to quality improvement as a result of exchange of genetic material within the bodies of living organisms, especially during gamete formation in prophase 1. That is where crossing over occurs, and therefore leading to variation and thus quality improvement. That is good. What is the other reason why reproduction is important? That was good. Yes, Boniface?
It leads to rise of a new offspring.
Leads to rise of a new offspring. Okay. That is a good one, but it doesn't bring really a point. It's like you're defining what reproduction is. Yes, I can see Sami Fulmo.
To procreation.
Sorry?
It leads to procreation.
It leads to procreation. And why would we procreate? Because you're still kind of saying the same thing. Why would we procreate, Sami?
Continuity.
So, yes, so we talk about preventing extinction, or we ensure continuity of living organisms. And those are the two main reasons why we carry out reproduction. Those are the two main reasons why we carry out reproduction. I see Leon Lewy's hand is up. Yes, Leon.
Leads to variation.
Yeah.
That one we have just said, it leads to variation. Yes, quality improvement, that is still variation. Thank you. So after that, I'd like us to note that there are various types of reproduction. So we have sexual and asexual reproduction. How would we differentiate between the two? How would you differentiate between asexual and sexual reproduction? Someone, how would we differentiate between the two? And if possible, you can give examples where those occur. Yes, I can see Boniface is very, very active. Yes, Boniface, please speak.
Asexual reproduction does not involve use of gametes, while sexual reproduction uses gametes. An example of asexual reproduction would be molting, and then an example of sexual reproduction is like in humans.
That is a very good one, Boniface, but your examples will make your answers not to withstand. Molting is not asexual. Molting is not asexual, but that difference you've given is very, very right. Though I would want a definition of what asexual reproduction is. I know sexual involves fusion of male and female gametes, but I want us to please define and not give an opposite of what asexual reproduction is. How would we define fully what asexual reproduction is? Yes, Florian. Florian? Florian, you can unmute and speak. Seems he is not there. So the definition that I would want us to embrace is that for asexual reproduction, we talk about a mature organism develops a new, please underline the word new, and a free living organism. A mature organisms develops a new and a free living. So please underline the word new and free living. Why? This is the basis of most, when we talk about asexual reproduction, they majorly depend on mitosis. That is to simply say that a unicellular organism, or just one cell, will divide mitotically and give rise to another new and free living organism. An example for asexual reproduction include one, binary fission in bacteria, or also you can talk about in amoeba. Then we have spore formation, or what we can also call as sporulation in rhizopas or bread mold. And then we have budding in yeast. Those are examples of asexual reproduction that we need to be very, very aware of. So a mature organism gives rise to a free and a new living organism. While, of course, sexual, we all know how to define, and I did define it. So let's move forward. We talk about now cell division. So the diagram below shows a phenomenon which occurs during cell division. Name part labeled T, and the biological importance of part labeled T. So can we get responses for number 2? A, B. Yes, I can see Evans is here. Evans is, so that is very good, Evans. Please unmute and speak.
Part label G is the chiasma.
So we talk about
Chiasma.
chiasma. That is very good. What is its biological importance? Evans.
It is where crossing over occurs.
The
It's
whole
where crossing
phenomenon
over
is,
occurs.
the whole phenomenon is called as crossing over. That is very good. I don't dispute. But what is the significance of that crossing over, Evans? Before maybe you leave. Why would we cross over?
It is where the chromosomes exchange DNA.
Okay, now
Chromosomes exchange DNA.
That is very good. Now chromosomes exchange DNA. What is the importance of these chromosomes exchanging DNA?
It leads to variation.
Now that would be the very, very right response. So here we have chiasma, and please note that this is just one. And then the biological importance is that it leads to variation. How? Because as a result of crossing over, there is the exchange of genetic material, which therefore leads to variation. What we were saying or talking about to mean quality improvement as a result of crossing over. Identify the type of cell division in which this phenomenon occurs. Yes, these ones tunafaa kuanguka nayo haraka haraka. Identify the type of cell division. Yes, 268.99, please unmute and speak. (268.99), unmute and speak. Seems that like he doesn't want to speak. Someone else? Oh, he's here. Okay. Thank you.
Meiosis.
That is meiosis. How many types of cell division do we have? 268.99. How many types of cell divisions do we have? Seems he has gone MIA. So we have, okay, him. Yes, him. Please unmute and speak. So we have only two main types of cell division, that is meiosis and of course mitosis. So this one is meiosis, and this is in meiosis 1 where we have prophase 1 in which crossing over occurs. Question number 3. The figure below represents cytoplasmic division in an animal cell and a plant cell. Identify the phase of cell division. And number 2, give 2 reasons that qualify illustration 1 to be an animal cell. So can we have someone answering the 2 questions simultaneously? Identify the phase of cell division and give 2 reasons why that illustration 1 is referred to as an animal cell. So can we have responses for those? Yes, anyone, any trials? Let us wake up and demystify this content and knowledge. Any responses? Identify the face of cell division. Yes, nonchalant aura. That's a very, very funny name.
It is a telophase, and that is an animal cell because it has like only a cell membrane.
Okay, for the first one, that is very good, nonchalant. But for the reason that you're giving, is a cell wall visible somewhere, or why would you talk about that reason?
I'm saying it's an animal cell because it has like just one. It has only a cell membrane. Does not have a cell wall.
Okay, but I'm asking you, please. When you look at illustration 1 and 2, is there any feature showing that this one has a cell membrane and a cell wall, while this one is just having either of the structure, from what we can observe?
Structure 2.
Yes, please.
Structure 2 is a plant cell because we can see the structure surrounding it is very rigid. It means it's a cell wall because the cell walls are very, very rigid.
Okay,
Yeah.
okay. Thank you for that. Thank you for that. That is a very good trial, but it won't qualify. Thank you for that good trial. Yes, Benson Karaoke. Please unmute and speak.
Can say the first diagram is an animal cell constriction of cell membrane.
That is very good. There is the constriction of the cell membrane. Any other reason? That is one reason. Any other?
And there's a formation of a cell plate in the cytoplasm of the second one for a plant cell.
So, Blesson, if I ask you,
yes,
give features of you that distinguishes you from me, would you be talking about you or me?
Me.
Okay, that is answering what you are trying to tell us. You are trying to tell us what feature 2 has that is not in feature 1. So what does feature 1 has that makes it to be called an animal cell? Yes, that is it is there. It is there. Just look at it well. 26 to 62, 76, please unmute.
Presence of centrioles.
And that is very good. Kingdom plantae lack centrioles. And that is the only kingdom where centrioles are not present. So we talk about the presence of centrioles which are used in cell division. And of course, name structure labeled T. Now, part C of that question. What is structure labeled T? Blesson?
Yes.
What is the
structure T is a cell plate.
That would be a cell plate. Then just proceed. What is part D? Can you answer question D? Give 2 reasons that, name the part of the plant from which the cell 2 was obtained. I think Blesson. Yes, 26899.
Kindly pardon the explanation.
For 3B, okay, for 3B, we are talking about the constriction of the cell membrane. For animal cells at telophase, they constrict the cell membrane. And then, additionally, we can see there are centrioles here, which are lacking in this plant cell. So for that reason, we say that this cell 1, it has those centrioles. I hope that now makes sense.
Okay.
All right. So part D, name the part of the plant from which the cell labeled 2 was obtained. And we need to move faster. We need to move faster. So from which part was cell level 2 obtained? So to answer that, because I see no one is going to raise up their hand. Okay, there is someone here. 26759, kindly unmute.
The answer, is it the root tips?
That is very, very close, but quite specific. Maybe you can talk about meristemic tissues, because those are the regions where we have cell division occurring. Because when we talk about this is basically the end of cell division, and in plant, growth always occur at specific regions, and those which we call them as meristemic tissues. And then, of course, we have apical meristems at the apex, and the cambium meristems in the stems of dicotyledonous plants. So at what stage of meiosis is chiasmata formed? At what stage of meiosis is chiasmata formed? This, I think I've just talked about it. Yes, there is Evans. Evans, at what stage is that formed?
Phase 1 of meiosis.
That is good. The significance of the above part in the living organisms is, of course, the significance of chiasma formation is bringing about genetic variation. Then part B, man 2, state true importance of meiosis in living organisms. Importance of meiosis in living organisms. I want to see new hands up. Are other people sleeping or what? Yes, we have Victor. Victor, please kindly unmute and speak.
It occurs in reproductive cells, leading to gamete formation.
So gamete formation, any other? There are so many significance, about 3 of them.
There is chiasma formation leading to variation.
That is also very good. And lastly, so we talk about gamete formation. Of course, it is the only process in the body of living organisms that leads to gamete formation. And then secondly, we talk about variation. Of course, there is the crossing over leading to variation. And lastly, we talk about it always halves the number of chromosomes present in the gametes. And why do we need those gametes to be haploid? Such that when fertilization occurs, we restore the diploid nature of living organism cells. So it ensures that the gametes contains half the number of chromosomes. And if asked to explain, you talk about it ensuring that we restore the diploid nature of living organism cells. What about the importance of mitosis? Why is mitosis important to living organisms? Yes, I need to see hands up. I saw Churi here. Yes, Churi. Good. Right on time. You can unmute and speak, Churi. Please, okay.
It is the basis of sexual reproduction.
Yes, give us about 4 importance. Sorry, yes, importance or significance of mitosis.
Okay. Let me start again. It leads to growth of an organism. It is the basis of asexual reproduction. It ensures that the chromosal constitution of the offspring remains the same as that of the parents, and it also leads to replacement of damaged or dead cells.
That is very, very good. Thank you for that, Sachiri. So mitosis is very important because it leads to growth. That is, should be number 1. Apart from growth, it leads to healing and repair. Then it maintains the chromosome number. While meiosis halves it, mitosis maintains the chromosome number. And of course, it is the basis of asexual reproduction, like binary fission, what we were talking about, We talk about budding in yeast and, of course, spore formation. List 4 differences between mitosis and meiosis. 4 differences between mitosis and meiosis. Yes, Chris Savio. I believe this is from Duke. Yes, Chris Savio.
Mitosis occurs in one phase, while meiosis occurs in 2 phases.
That is good. Give us more differences so that we don't spend time asking about others also. What are the other differences between mitosis and meiosis?
That's the only one I got.
Okay, no problem. Thank you for that. So mitosis occur in 2 phases while meiosis occur. Sorry, mitosis occurs in 1 phase while meiosis occurs in meiosis 1 and 2. Yes, Mutiso, give us more.
There's no crossing over in mitosis, but there's crossing over in meiosis.
That is very good. Any other? Give us more. Give us more.
Mitosis occurs in the body cells, and meiosis is for the sex cells.
Okay, so mitosis occur in somatic cells while meiosis occur in the gonads, so the sex cells. Any other?
I don't have any other.
Okay, that is good, Muti. So thank you for that. There are more. Yes, yes, I can see him. Him is back again. Yes, him. Him doesn't want to unmute. I can see Boniface. Boniface.
In mitosis, the number of chromosomes is retained, while in meiosis, the number of chromosomes is halved.
That is good. And also we have, in mitosis, we form 2 daughter cells, and in meiosis, we form about 4 daughter cells. All right, let's proceed. I think for cell division, we have done quite enough. I think this is the last one. We can do number 7, 8, and 9 for the last one so that we can move to another subtopic. So for number 7, we talk about the diagram below represents a stage in cell division. Name the stage of all division, and this should be cell. Name the stage of cell division shown in the diagram above. Which stage is this of cell division shown in this diagram? And give a reason for your answer. Any trials? Yes, I have Blesson Karaoke here. Yes, sir. Please unmute and speak.
The cell stage is metaphase 1.
Give a reason for your answer.
Since there's homologous chromosomes aligned at the middle of the cell.
Okay, metaphase 1, homologous chromosomes aligned. Why would you say it is metaphase 1? Because of homologous chromosomes?
Yes.
That one I wouldn't agree to. That very, very well. Thank you for that. So this is just metaphase, and the reasons we give is maybe just so to give an overview of all these types of cell division, of these stages of cell division. Of course, we have the acronym, or how do we call that? Yes, we have this word called as IPMAT to give us the 5 stages. And maybe just so to emphasize, please let us note that at interface, whatever occurs at interface is very, very important or significant to what will happen in telophase, the last phase. And what do I mean? Remember, when we talk about cell division in biology, it is the only concept in life where we talk about division to mean multiplication. How? In mitosis, for example, assuming we take an human cell that is having 46 number of chromosomes, let us note that in mitosis, we need to have at the end 2 daughter cells and each daughter cell must be having 46 number of chromosomes. So how do we get 246 and yet we had only 146? It is because at interphase, whatever we do is going to give us whatever we have in telophase. How? At interphase, we multiply the DNA. So the chromosome number is doubled, or what we call as DNA replication. 2, because we are going to have 2 new daughter cells, organelles are very, very important, especially, for example, like mitochondrion. It is used as the powerhouse of the cell. So for the 2 daughter cells, each must have its own mitochondrion or organelles in general. And as a result of that, the first thing that occurs is DNA replication. And secondly, we talk about synthesis of new organelles. And lastly, the third most significant thing that occurs in interphase is building up of energy that is required for cell division. And those 3 things, they are very, very significant when it comes to telophase, the last phase, because we are going to divide those cells to have 2 daughter cells, and each must have 46 number of chromosomes. Each must have new organelles. And of course, that energy is required for cell division. Then followed by prophase. At prophase, what do we say? Remember, during DNA replication, we have the opening of those chromosomes. So they appear thread-like at interphase. But at prophase, they need to condense to form the chromosomes again. So they will condense. And at early prophase, you'll be having them still within the nuclear membrane. But at late prophase, I always call prophase as that type of cell division phase where tunamwaga kila kitu ndani ya nucleus inside the cytoplasm. And how is that done? The nucleolus will disappear, and its disappearance is what causes the nuclear membrane to disintegrate. And as a result of that, tunamwaga every content in the nucleus inside the whole cell that is inside the cytoplasm. And then at metaphase, what is in question here? We go fishing now. We go fishing. So you will learn again that at late prophase, the spindle fibers have begun to form, meaning the centrioles, they had migrated to the opposite poles. But at metaphase, we've gone fishing, meaning the spindle fibers have attached to the centromeres, like shown in this diagram. They are attached at the centromeres of those chromosomes. And of course, metaphase means middle. The chromosomes are aligned at the equator or at the equatorial plane of that whole cell. Then, of course, anaphase is anakimbia. What will be running? Remember, we want to divide the chromosomes and thus the cell. So the chromosomes will be running towards the opposite poles. And what makes them to run or go away from the center is because of shortening of these spindle fibers. And of course, at telophase, we just now make or remake our eukaryotic cell. Why am I heavy on eukaryotic cell? Most plant cell and animal cells, their nucleus are bound by membrane. And as a result of that, when those chromosomes arrive at the at the poles, that is south and north, they have to be enclosed within the nuclear membrane. And as a result of that, the nucleolus will reappear. And that also makes the nuclear membrane to reappear. And that gives us a summary of what occur in those main phases. So I think having talked about that, you can at least go out of your way, look at mitosis and those significant events, meiosis 1, and of course, meiosis 2, which looks exactly like mitosis. So I think I'll skip that because they are the same things that we will be talking about all through. So I'll go to another subtopic so as not to waste on time. So the other subtopic, of course, is asexual reproduction. So let us look at question number 16. What is asexual reproduction? Responses.
Reproduction without combination
Yes, Florian, what is asexual reproduction? Florian, you can unmute and speak. He's not here with us. He's doing other things else. Okay, let Rose Wanjoike, can you unmute?
Production of offsprings without fusion of gametes.
Okay, that is a good trial, though I did say that let us not give opposite of what sexual reproduction is. Let us define, really define what asexual reproduction is. And I did define that in our class just now. Yes, I can see Trevor. Trevor, please unmute and speak.
It is where a free existing organism develops into another free existing organism.
So we did define it just this morning, and we said that a living organism that is mature will develop a new, please underline the word new, and a free living organism from itself. And there are types of asexual reproduction that occur in living organisms, and we have budding in yeast, spore formation in rhizopas, and of course we have binary fission in amoeba, and that can also occur in bacteria. State the advantages and disadvantages of asexual reproduction. The advantages and disadvantages of asexual reproduction. Yes, any trials, any answer? Yes, Blessom. Blessom, can you unmute? Okay.
Good characteristics are taken to the offspring in asexual reproduction, the advantage. And also it does not take a lot of time.
That is good. What about the disadvantages? We'll give this to Abdul. Let us give this to Abdul. Yes, Abdul.
Rapid population growth. It allows rapid population growth. A single individual can produce many offsprings.
That is very good. Like bacteria bacteria always multiplies in a span of 20 minutes, and we have a new free living bacteria again. So there is a faster rate of development or growth of population. Then of course, bad characteristics or bad traits are always passed to the offspring. What is sexual reproduction, and what are its advantages and disadvantages? Yes, we have Trevor here, we have Blesson here, we have Abdul here. Yes, we have Trevor, yes.
It's the type of reproduction where it involves fusion of fertile male and female where it includes fusion of fertile male and female.
So we have fusion of female and male to form a zygote.
Gametes.
Okay, that is fine. What are its advantages and disadvantages?
In advantages, it leads to variation due to crossing over.
Yes, that is good. Any other?
That's the only one have.
Okay, that is good. Let us hear from others. Where are others? Yes, 26294. Please unmute and speak.
Introduction of useful traits to the offspring.
Introduction of useful traits in the offspring. Any other?
No.
Okay, what about the disadvantages?
It takes a longer time.
Why? Why would you say it takes a longer time? Anyways, it takes a longer time.
Then
I think we may proceed. I think we can proceed to question 18. A student carried out a study to observe the methods of reproduction and feeding in amoeba species. He observed that amoeba doubled its number after every 20 minutes. So in part A, name the type of reproduction shown by the species. Draw the process of reproduction in amoeba. Then describe the principle used in reproduction in amoeba species. Then state the function of pseudopodia in amoeba. I want someone to answer all these questions except for drawing. And for drawing, I'll talk about it. Yes, I can see Rose here. Start from A all the way to D.
The type of reproduction shown is binary fission.
Binary fission, good.
The principle used is when there is optimum conditions of temperature and pH, the amoeba species divides into two and continues splitting into two data cells that are separate from each other. The function of pseudopodia is for movement, and the type of cells in the human body which show a similar biological process are reproductive cells, like sperm cells.
Okay, that is a good one, though there are some places where you have not elaborated well, like part E and part C, but that is very, very good. Any other response from someone else? That is very good, Rose. So the type of reproduction is, of course, binary fission. You be specific. This is a sexual type of reproduction, which is binary fission. Then draw the process. In that process, when you are drawing it, please, the first diagram you draw an amoeba cell with only one nucleus. And then in the second diagram, you draw that same one cell, and then you show that there is karyokinesis. And karyon is the nucleus, and that is why we talk about eukaryotic and prokaryotic. So karyon is nucleus, so the nucleus will divide, and that is what we call as karyokinosis. Karyokinesis, and kinesis means movement. So the nucleus divides, and then in the third diagram, you draw when you show now what you call as cytokinesis. The cytokinesis is the division of the cytoplasm. So you draw the constriction, because this is not a plant cell, but an animal-like cell. I know it is under kingdom Protoctista, but you draw it having the constrictions of the cell membrane. Then for part C, describe the principle. Like Rose has said, there must be optimum conditions like food, temperature, and even pH. Then what happens first? The amoeba will retract the pseudopodia, those projections, so that it forms a rounded cell. After that, then the nucleus divide. Then after that, it is followed by division of the cytoplasm. And then you'll be having two identical cells. And that is what or how that is achieved. Then state the function of pseudopodia. Rose, instead of talking about movement, be specific. It is used for locomotion. And then for part E, name the type of cells in human that show the similar characteristic as shown in this organism. So for the characteristic that could be in question here, we could be having various responses. One, we can be having a response talking about in terms of reproduction. So if it is in terms of reproduction, we can talk about the liver cells. The liver cells divide very, very, very fast. That is number one. When we're talking about movement as another characteristic, because we have reproduction and again movement here. When we talk about movement, we can talk about white blood cells because they also have amoebic movement. They show, they squeeze through those semi-permeable pores of the capillaries and move into the area of infection. So that you can be answering it in two ways. For reproduction, you can talk about liver cells. Liver cells regenerate very fast. And then for movement, we can talk about white blood cells. I think for that, we can also move away from it and move to another part. So these are all about asexual reproduction. And I did say I will share these questions. Wilson has a question. Yes, Wilson. Wilson, you can unmute and speak. Okay, it seems maybe he raised his hand unknowingly. Let's proceed. So these are, again, these are other asexual types of reproduction you can also look at because this document, I will share it with you. Evans has a question. Yes, Evans.
May you pardon the last question you just explained, where the liver cells divide faster, the white blood cells move.
Okay, fine. So I was saying that when we look at what the question here is talking about, you can see that these amoeba cells, they divide after every 20 minutes.
Yes.
So the only cell that is capable of rapid regeneration after a very short time is the liver cells. They regenerate very, very fast. And then
yes.
Again, looking through the question, you understand that there is another characteristic of living organism that we are talking about here, which is movement. So if you talk about movement, the only cell in human beings that carries out that type of movement, which we call as amoebic movement using pseudopodia, is the white blood cells. So that can be asked in two different ways, and we need to be very specific. I hope that is now in order.
It's in order.
Okay, thank you. Wilson again has raised up his hand. Yes, Wilson.
Teacher, I wanted to say that, would you please pardon on the principle used in reproduction of amoeba species?
Okay, the principle that is used in reproduction in amoeba species, majorly, it is just trying to explain how mitosis occurs. Mitosis is the main basis of this reproduction. So what happens? When there are good or appropriate conditions of temperature, pH, and even enough sufficient food, remember mitosis. Why are we talking about mitosis in that order? For a cells to divide, it requires glucose. For cells to divide again, it requires oxygen because it's used in the production of energy. So now, if all those conditions are met, then the amoeba will retract the pseudopodia, those projections that it uses for movement. And then after retracting those pseudopodia, then you will be having mitotic division of the nucleus. That is what I'm referring to as karyokinesis. Karyon is just the nucleus. That is why we talk about eukaryotic and prokaryotic. Then, after you divide the nucleus mitotically, then the cell membrane begins to constrict, just like that which we find in animal cells. And after that, you are going to have two daughter cells, each having the same chromosome number as that of the parent cell that you had. That is what we are talking about. I do believe that is in order now. Wilson? Is that in order? Very good. I can see you are saying that is very, very okay. So there are these other questions on reproduction again, asexual that you can do after maybe you receive this document. Then the other part is about the flower. So about the flower, we talk about these terms, epigynous, staminate. Can someone define those terms? Yes, I can see Trevor. Trevor is very, very active. I don't know where others are sleeping.
Epigynous flower is the flower where its ovaries located below other floral parts.
Epigynous below. Okay, that is a good trial, though not really, Trevor. Yes, Victor Kinuthia is gone again. Why is he gone? Okay, Victor has gone. But well, epigynous, epigeo means above the ground. So epi always means, it's a prefix used to mean above. So when you talk about epigynous flower, it means that, by the way, what is epigyny? What is epigyny? Yes, Derek, what is epigyny? Derek is also going away. These people are going away now. Well, epigyny refers to floral parts in relation to the ovary. Like in a flower, we have three types of epigyny. We have epigynous flowers, then we have perigynous, and of course, hypogynous. And epi means above. So means that the floral parts, they come above, or they originate from above the ovary. If my head is the ovary, if you can see me, then if the sepals, the petals, they originate from above the ovary, that is what we call as epigynous. Then if they originate from below the ovary, that would be called as hypo, just like hypotonic solution, hypogel type of germination, below. Then peri, of course, it's at the periphery, the same level with the ovary. So they will be at the same level with the ovary. Then a staminate flower. What is a staminate flower? 26899. What is a staminate flower?
Okay, I wanted to clarify because for the first question, epigynous, he mentioned that the ovary is located below the floral part. So basically, that's the same thing.
Epi means the floral parts are above. Okay. So the ovary is
So that
below.
means the ovary is below.
Thank you for that.
So you can
About a
explain
staminate
it like
flower?
that.
Yes, you can.
Okay,
What
staminate
about
is a
yes,
flower. It's a flower which contains only male.
yes, so it's a flower that only contains male parts, and that is what we call a staminate flower. And we have two main types of unisexual. Uni means one, like unicellular organisms. We have two types of unisexual flowers. A staminate flower only having the male parts, while a pistillate flower having only the female parts. The diagram below represents two gynoaceae, A and B, obtained from two different plants. Name gynoaceae A, name gynoaceae B. State the observable differences between gynoaceae A and B. Yes, who will answer that for us? Chiuri went away. I don't know where Chiuri went to. Where are others? Boniface is also quiet. Yes, 26899. You are also very, very active. Please unmute and speak.
Gynoecium A is monocarpous flower. Then Gynoecium B is polycarpous.
Monocarpous. Then okay, that's a good trial. Thank you for that. Someone else? Yes, I have Ross.
I agree with monocarpous, and then Gynoecium B is apocarpous flower.
That is also very, very good. I need someone of a different opinion. 286.
Genuitium A is Cynocarpus, while Genuitium B is Polycarpus.
Syncarpus and polycarpus, and that is, I think, the right response. Please look at this. We have two types of, okay, when you are defining or describing carpels, we have monocarpus, and monocarpus is just only one, a flower with just only one carpel. And what is a carpel? One stigma, one style, and one ovary. That is monocarpus. Then we have those flowers that have several carpels, and those we call them as polycarpus. So in polycarpus flowers, we have syncarpus where the carpels are fused like in A, and then we have apocarpus where they are not fused like in B. And those are the only types of carpels that give us parietal, free central, and axile type of placentation, where we have the carpels occurring in groups. There are many. But when you talk about monocarpus, monocarpus only gives us two types of placentation. That is basal and marginal. So the observable differences is that in A, they are fused, in B, they occur singly. They are one on each other. So they're all played by pterostylene plants. They're all played by pterostylene plants. Yes, Chris Avio, you can unmute.
Heterostyle hinders self-pollination.
How?
It is where the stigma is above the anthers so that the pollen grains don't reach them.
That is very good. So hetero means different and style is the style. Of course, the style grows longer than all other floral parts, thus ensuring that the stigma is far much above the male parts of the stamen. I see Kip has a question. Kip, you can unmute and speak.
Can you say it enhances cross-pollination?
Also, that is a significance. It enhances cross-pollination by hindering self-pollination. I think we can move to another part. All those other parts, again, are just about flowers, and you can also look through them. If maybe you get stuck somewhere, you can ask friends, or you can always reach out to me to help you answer those other parts. So we can move to the other part, that is pollination and fertilization. So for pollination and fertilization, we are being asked to name the part of the ovule that develops in each of the following parts. We have tester and endosperm. Yes, 2, 6, 8, 9, 9.
Okay, for the tester, it's the integuments.
That is okay. What about the endosperm?
The endosperm is the which from the uses the polar nucleus. So they form the primary end of that develop into the endosperm.
That would be the triploid endosperm. So the triploid endosperm is what develops into an endosperm. Part B, what is parthenocapy? And this is how I'd want to ask this question. Differentiate between parthenocapy and pathogenesis. Differentiate between parthenocapy and pathogenesis. Where are others? The same hands are up all the time. Where are others? Yes, Holistic Enterprises. Please unmute and speak. Holistic Enterprises, unmute and speak. Maybe is not here with us. The same hands up. 262.76. Okay, 262.76. You can unmute and speak. He doesn't want to unmute. I don't know what is wrong. Yes, Larry, please unmute and speak.
Could you say parthenocarp is a fertilization of organisms without the fusion of gametes?
Larry, if you talk about fertilization and then you talk about without fusion of gametes, you are really, really contradicting yourself. Give it another trial, please.
Okay. It is the formation of a zygote without formation of fruits without the fusion of gametes. Okay, what I do know is that there is no fusion of gametes.
Yes, now that is very, very right. Formation of a fruit without fertilization. That is very, very right. But, and what is about what about pathogenesis? Yes, Raviz. Unmute and speak, Raviz.
Pathogenesis is reproduction by development of an unfertilized egg.
Yes, the development of an organism with the development of an unfertilized egg. Can you give as an example of where that occurs in maybe in animals?
For example, in certain species such as fish.
In fish, not really, not really. That is a good trial, though. That is a good trial. Let's have Dora Okwaro saying something. Dora? Yes, Dora Okwaro. He doesn't want to unmute and speak. It is okay. So pathogenesis occurs in animals while parthenocarpy occurs in plants. And as a result, we talk about parthenocarpy to mean that process whereby a fruit develops without fertilization. And for pathogenesis, we can have eggs hatching into living organisms without fertilization. And that occurs in fruit fly, aka Drosophila melanogaster. Why is cross-pollination more advantageous to plant species than self-pollination? Yes, answers yes. You please unmute and speak.
Mwalimu, could you repeat pathogenesis?
All right. Pathogenesis, I am trying to say that it means the process whereby an egg hatches into a new living organism without fertilization. It is just opposite of what parthenocarpy is. It's only that now it occurs in animals. And a very good example is fruit fly. Fruit fly. The scientific name of fruit fly is called as Drosophila melanogaster. And that is what, when you open up next time, you will do so many of it or about it in genetics. Drosophila melanogaster. It is one of the model organisms that is used in biology to conduct genetical experiments. Any other question? All right. So why is cross-pollination more advantageous to plant species than self-pollination? I hope you got that right. Yes, Raviz? Raviz is gone, MIA. Yes, we have Victor.
Cross-pollination leads to variation of characteristics.
That is very, very okay. Let us proceed to question number 27. The diagram below shows a pollen tube as it develops down the style. Name the parts labeled M and N. Name the parts labeled M and N. Yes, I can see Sami Fulmo is back. Yes, Sami?
For the part marked M, we have the male nuclei, then N, the tube nucleus.
That is very, very good, Sami. State the functions of part labeled M. Someone else. And that gives us the definition of what double fertilization is. 26899, please speak. You already yes, please.
Okay. So the male gametes nuclei, they fuse with the egg cell and the polar the and the triploid nuclei.
I didn't hear that quite fine, but I do believe, okay, let us also hear from Reni. Reni has been quiet. Yes, Reni, unmute and speak. Reni, you can unmute and speak. Or Reni is like, is not here with us. Jeremy Jeff, please unmute and speak.
They fuse with the egg and the polar nuclei to form a diploid zygote and a triploid endosperm zygote, respectively.
That is very, very good. So one male nucleus of the generative nuclei will fuse with the egg cell to form a diploid zygote, and that would be the first type of fertilization. Whereas the other male nucleus will fuse with the polar nuclei to form a triploid endosperm. State role functions, okay, that one we have done. So these are the same kind of questions for that same particular part. That is all about double fertilization. So we want to be doing the same kind of things all through. We have now fruit and that dispersal. I want to skip on that and look at something about animals, maybe before our time comes to an end. I see we have about 19 more minutes to go. So let us talk about sexual reproduction in animals and maybe do a few questions before the time is up. So first question is asking, distinguish between external and internal fertilization. What is the difference between external and internal fertilization? And please, for each, give the classes of phylum Chordata whose members show those type of reproduction. So who will give us the difference and give us example of the classes in whose members those types of fertilization occur? Yes, responses. There is Jeremy, there is Boniface, there is Fulmore, there is, yes, there is a 26108. Please unmute and speak.
So the difference, to distinguish external and internal fertilization, can say that external fertilization is a type of fertilization that takes place outside the female body, while internal fertilization takes place inside the female body.
Very, very well. Can you now give us examples of those various classes in which these types of fertilization occur?
So for external fertilization, we have class external, we have class Amphibia and class Pisces.
So we have class Pisces and class Amphibia. And what about in internal type of fertilization?
So internal, we have Mammalia, we have Reptilia, and we also have Aves.
And that is very, very good. Thank you, 26108. I wish I knew your name. Next time, maybe join using your name. That is very, very good. So of course, fertilization is defined as fusion of male and female gametes to form a diploid zygote. But these can occur either internally or externally. So for external, we talk about the fusion of male and female gamete occurring outside the body of a female organism, like in most fish and even amphibians. So we have class Pisces and class Amphibia. Most fish and even frogs, the male will always mount on the female, such that when the female is shedding those eggs, then the male again will be, so if the female is laying those eggs, then the male will be lying over it, shedding those eggs. What about internal type of fertilization? For internal, of course, the fusion of male and female gametes occur inside the body of the female organism. And then here we have birds, We have human beings, of course, that is Mammalia. So we have aves, class Mammalia, and of course, class Reptilia. And maybe just so to expound something a little bit, after fertilization, especially, which is internal, we have three various types of embryo development. And this, please write them down. We have three various types of embryo development. One, oviparous or oviparity. Two, oviviparity or oviparous. Three, we have viviparity. What is the difference between the three? So this one, please note them down. One, those that are having O's, please note that the egg is formed. So that is going for oviparous and oviviparous. So the egg is formed in both of them, the ones that are having O's, oviparous and oviviparous. Then again, another note, as you are defining them, writing them down. If an egg is formed, it means that the embryo will get nourishment from the egg yolk. So the embryo gets nourishment from the egg yolk, both in oviparous and oviviparous. Then leave that at that. Let's talk now about viviparity or viviparous. So for viviparity, vivi means inside the body of the organism. So we have these terms called as in vivo and in vitro. So when you talk about vivo, means inside the body of a living organisms, while in vitro means in the laboratory. So if vivi means inside, then viviparity simply means that the embryo develops inside the mother's body. And for viviparity now, the nourishment is gotten from the mother directly, meaning that baby gets the nourishment from the mother directly, as in mammals. Then when now we go back to oviviparity, it also has that word vivi, and therefore it means that even though an egg is formed and the embryo gets nourishment from that egg yolk, this egg will stay in the mother's body until the time for parturition comes, whereby those species or those organisms, they will be giving birth to live young ones. Like in species of snakes, there are those snakes that will keep their eggs inside their bodies until the time reaches where they will be giving birth to live young ones of the same kind. So that gives us the three types of embryo development. Oviparous, kuna mayai, there is an O. Oviviparous, there is mayai, and then there is vivi again. And vivi means inside the mother's body. And lastly, we have viviparity, and that simply refers to that situation whereby the baby, develops inside the body of the female organism. And as such, those are what we have. All right, let's proceed. I think for part B that has been answered. That would be phylum, the classes would be class Amphibia and of course class Pisces. So the next question is asking, give a reason why frogs have to lay many eggs. Yes, BK Kariuki.
This increases the chances of survival in the offspring.
So that increases the chances of survival. Any other reason why they need to lay many eggs? Yes, Dora Okwaro.
Okay, increasing others may fail to be fertilized due to bacterial attack and predation.
Yes, others may fail to be fertilized because that again will depend on chances. And why do human beings have to produce so many spermatozoa, and yet only one is going to fertilize that egg? Yes, Dora, still on you.
Okay, because okay, the vaginal tract may there will be death of the sperm. Hello.
There are so many reasons for that. There are about 5 or so reasons why more sperms are supposed to be produced. Yes, Mutiso.
The acidity in the vaginal tract, and acidity also in the urethra. Also, some of the sperms may end the energy, they may be depleted of the energy.
Yes, two more reasons.
I don't have any more reasons.
Those are good. So there is the pH variations both in the urethra and also in the vagina, and then there is the long distance. So others will maybe deplete their energy. Yes, you please unmute and speak.
Mwalimu, can you say that the excess sperms that don't get fertilized are used for nutrition to the egg?
No. For nutrition to the egg, no. The egg gets nourishment from the follicle cells. That is something that you must get very, very right. And secondly, please, there is no digestive system in a reproductive system. You can never find that. So it is not true. So the other 2 more reasons we can give is or are inappropriate temperatures for manufacturing or manufacturing temperatures for spermatogenesis, and of course inappropriate temperatures for storage. So those are the 2 other reasons that we can give, apart from the long distance they have to cover and others will deplete their energy. And of course we can talk about barriers like the cilia present along the oviduct, because those cilia, they again will waft to move the egg away. So if the sperms are going towards that direction, they will meet such barriers. Let's proceed to the next question. I see time is almost up. We only have 8 more minutes. So I'll do one more question and then you will be free to ask questions. So question number 51. State the functions of the following parts in the male reproductive system: seminiferous tubules, and of course Sertoli cells. And that is where we are going to end. Yes, 26670, please unmute and speak. Yes, 26670, please unmute and speak. 26670, you can unmute and speak.
Seminous ferrous tubules, which provide a large surface area for sperm production, and Sertoli cells that nourish the sperms.
That is very, very good. Now, I think we'll need to end our discussion there. And if someone is having, if you are having questions, please raise up your hand and speak. You can raise up your hand and speak if you have questions. So we start with Trevor. I see is then Psionic. Maybe you can unmute and speak.
Kindly, can you pardon the points, the answers to the previous question?
Question 51, or which one are you talking about?
Yeah, question 51.
Good. So the testis has 3 main functions. 1 is to produce sperms, 2 is to nourish them, and 3, we talk about production of hormones. So respectively, we have seminiferous tubules, Sertoli cells, and interstitial cells. So for seminiferous tubules, these tubules are long and highly coiled, and that offers an increased surface area for sperm production. while Sertoli cells secrete an alkaline fluid, which is used to nourish those sperms. And lastly, we have interstitial cells, whose main function is to produce hormones, and male hormones are collectively called as androgens. And a very good example of androgen is the testosterone. Yes, Evans, I think you are answered. Evans?
Would it be right to refer to the Sertoli cells as giant Sertoli cells?
Giant is a
Would
descriptive
it?
term.
So it's just okay.
It is okay, just very, very okay when you talk about Sertoli cells. Thank you. Yes, Raviz? Please unmute and speak.
Could you pardon the points on why the sperms are needed to be secreted in large numbers?
All right. Point one is that we have inappropriate storage conditions there in the testes and also inappropriate manufacturing conditions. That is one. Number two, the distance that they cover from the epididymis all the way to the fallopian tube is quite long. So others will deplete their energy on the way. Three, we talk about the pH variations caused by urine, both in the urethra and also in the vagina. Those are the main reasons why, even though only one sperm will fertilize that egg, we need to produce so many of them. I believe you answered. We have 4 more minutes to go. Yes, Dora?
Can you also say the egg produces a chemical that inactivates the sperms?
Yes, that can also be said. But yes, that can also be said. I don't want to talk about the but now. It can be said. Yes, you?
Oh, Mwalimu,
Yes, please, sir.
I wanted to ask, why is double fertilization in flowering plants considered an evolutionary advantage compared to the fertilization process in animals?
That is a very good question. So the response to that is, when you look at fertilization in human beings, we have an haploid sperm cell and an haploid ovum cell giving us a diploid zygote. But in plant cell, we have the triploid endosperm being formed. So meaning there is an extra set of chromosome that is given. And what you are asking simply, is what we refer to as polyploidy. Polyploidy in genetics is an advantageous in plants because of the following reasons. One, it ensures resistance to diseases and pests in plants. Two, it ensures or encourages high yield production in plants also. So those are the reasons why it is referred to as an advantage in plants rather than in animals. And the last question is from Dora Okwaro again. Please unmute and speak because I still can see your end up.
Okay. The reason why the fertilization must take place in the fallopian tube and not in the uterus.
Okay. Why must we have fertilization taking place in the fallopian tube and not in the uterus? This is the reason. That egg after ovulation, it takes about 8 to 24 hours before it dies. And then for it to travel all the way to the oviduct or the womb, that takes about 7 days. So once it is produced, because of the short time it takes before it dies off, that is 24 hours, a maximum of 24 hours, that fertilization must just take place there in the oviduct. I believe you have answered. Thank you for listening, and I will share this with you through your form coordinator. Have a good day ahead, and bye-bye. Thank you, bye-bye.